Hello, my name is Shaheen Ahghar. I am a paramedic and I have a diploma in mountain medicine. I'm here to tell you and talk to you about something important to me today. It's called acclimatization. It's a riveting story of partial pressures, pH, and hypoxia inducing factor. Now, let's go and talk about the overview. Acclimatization is the physiological response to hypobaric hypoxia. The response takes place within minutes to weeks and involves multiple organ systems. Now, hypobaric hypoxia is when barometric pressure decreases with ascent and the partial pressure of inspired O2 decreases along with it. The pressure gradient decreases in the alveolus, causing increasingly inefficient gas exchange. This effect trickles down the oxygen cascade, resulting in progressing hypoxia. Now, let's talk about partial pressures of oxygen. We have this graph to illustrate what's going on at high altitudes. We have on the bottom altitude with rising elevation and partial pressures of oxygen, as well as the arterial oxygenation barometric pressure on top. We have the arterial oxygenation here in blue and the PiO2, inspired oxygen partial pressure, and the arterial blood O2 partial pressure on the bottom here. As you can see, as the elevation increases, there's a significant downward trend of all of these. And we have something that's really important that we'll discuss now because it makes such a big impact on all the rest of the organ systems, and it's called hypoxia inducible factors. HIF, as it's called, plays a massive role in the gene response to acclimatization, particularly HIF1 alpha, since it is the main HIF regulated by O2. Thus, HIF1 alpha starts the gene regulation cascade or it ends it. Research on HIF's oxygen sensing ability won the 2019 Nobel Prize for Physiology and Medicine. Now let's discuss more a little bit. The HIF1 alpha, it regulates up to around 4,000 genes. HIF1 alpha is regulated allosterically by two proline residues that we'll talk more about that are hydroxylated under norm-oxic conditions, leading to eventual breakdown in peptides. In the absence of O2, HIF1 alpha enters the cell and combines with a HIF beta to begin gene regulation. And once it combines with that, it is just called HIF1. This leads to erythropoiesis and angiogenesis and metabolic changes. Now we see this diagram. We have the top, we have normal oxia. We have HIF alpha. It comes in contact with the O2 and becomes visible for the von Whipple-Lindau protein, which is a tumor suppression protein. It marks it for eventual recycling or termination, as you might want to call it. And you can see that it eventually becomes a bunch of little peptides in an oxygen-rich environment. But we have a hypoxic environment. You see there, it goes in the cell, combines with the HIF beta, starts that gene regulation, starts that erythropoiesis, metabolic changes, and angiogenesis. Now, this is a little more involved when it comes to the regulation, this diagram. The multicolored bar in the middle kind of represents the HIF1 alpha. And you can see towards the middle, we have two little bumps. Those are the proline residues, and we can see it's in a hypoxic environment, and thus the VHL does not bind onto the proline residues. And what happens with an asparagine residue in this hypoxic environment, it attaches to P300 CBP, which is a transcription activator, so it ramps that up. In normal oxic conditions, we have O2, and it starts to bind with the proline residues and hydroxylate it. Alpha-ketoglutarate comes in as a substrate with the O2, which is eventually rendered into succinate and CO2. And the asparagine also gets in contact with the O2, and the P300CBP is jettisoned off, and the VHL attacks onto the proline residues and starts the eventual recycling. Now, there are a few types of HIF 1 alpha, and they're all in the same cells, but HIF1 alpha occurs in all nucleic cells. HIF1 2 and HIF1 3, only some cells in vertebrates. Now, HIF1 or HIF can go bad and cause mutations, namely congenital polycythemia, which is a clotting disorder, and is caused by the mutation pathway for HIF to be inhibited, or the mutations for the inhibition of HIF pathways causes it to overproduce and thus cause all this extra matter in the blood and thus causes clotting disorder. Now we're going to go and talk about the respiration blood gases and renal changes. They're very interwoven, and the first to kind of respond to acclimatization. The increased respiration is because there's less available O2, and the oxygen cascade is now inefficient, and as a result, hypoxic drive triggers a ventilatory response. Now we have alveolar CO2, which is lowered due to the increasing rate of gas exchange caused by the increased respiration. Thus, all the CO2 is blown off on the exhale, and carbonic acid is thus lowered, and because it's an acid, blood pH rises and alkalinity sets in. Now we have ventilatory compensation limit. The weakest link is the brain. It has central chemoreceptors in the medulla that sense the pH, and when it gets to a point where it can't go any higher, it can't stand it anymore, it downregulates the respiration. It kind of slows it down so the pH doesn't go any higher. Now, the renal system comes in handy, and the kidneys kick in and start excreting bicarbonate, being basic that is, and it starts lowering the pH. The lowering pH allows for more room for the respiratory compensation. And this usually occurs within 24 to 48 hours. Now we have the hematological changes as well. The increased hemoglobin concentration, and as well as increased oxygen carrying capacity of blood. And after a few hours of hypoxemia, renal cells stimulate erythropoietin, leading to red blood cell synthesis. Now we can go look at the oxyhemoglobin dissociation curve, and we can see that the pH on the left here is increasing and thus causes a leftward shift. And the 2,3-DPG is also elevated due to the environment that it's in, and thus causes a almost neutral shift where almost nothing happens. It keeps in balance and doesn't move unless in very, very high altitudes the pH eventually overcomes the 2,3-DPG. Now, we're going to be looking to circulatory changes. This includes cardiac output, heart rate, venous tone, and blood pressure. Carbonate diuresis also adds to the stress of the heart because it's losing all this fluid, and thus the heart rate increases. Cerebral blood flow increases to better oxygenate the brain under this hypoxic condition. We gotta have priorities. And we come to oxygen delivery, and the HIF1 comes in handy, and it starts stimulating vascular endothelial growth factor, VEGF, which stimulates angiogenesis and nitric oxide synthesis. And the nitric oxide vasodilates, resulting in a greater blood flow and perfusion. Now that about wraps it up. All we have left is my references. Those kind people.